Hey everyone, let's talk about protists today, and in this video we'll cover a lot of ground, but really I'm not going to cover everything in the PowerPoint, so everything that's not covered in this video, as in always all the short lecture videos, they're not meant to cover everything, so make sure you're reviewing the PowerPoint for the rest of the information, but this is the gist of what you need to understand for protists. So let's dive into it. So first things first, where are we at on the tree of life? Right, we have talked about bacteria and archaea. Now we're climbing up the tree, right? Not in time, just in terms of ancient, like how far apart we're the common ancestor. Now we get the common ancestor of all eukaryotes, and that's the common ancestor to all protists, which tells you a lot about that group. Already before, you should have realized that the prokaryotes are a paraphyletic group. Their common ancestor is also the common ancestor of other groups that are not called prokaryotes. Even if from all the tips of the trees of our prokaryotes you go back to only that common ancestor and you stay in the group of prokaryote, that's true in one way. The other way is not. From a common ancestor, you stay in prokaryotes, leave prokaryotes, right? Similarly, protists are a paraphyletic group. Their common ancestor, all of the branches go to the same common ancestor, staying in protists. However, from the common ancestor of all protists, if we go down all the tree branches, we get to animal, fungi, and plants. Other eukaryotes share the same common ancestor, so it's a paraphyletic group, which tells us important stuff. It means they're gonna be sharing really important characteristic with the different groups of eukaryotes that are also sharing the same common ancestor. So you have a common ancestor of plants and some of the protists here. Those will be more green algae or algae in general, but green algae are the one that leads to plant. Other algae types can be other branches of protists, right? Slime molds, water molds, it's more about the fungi. So protists that led to fungi are more of a slime mold, water mold type, and protozoa are more animals. So it's that they share common ancestor, right? None of the protists that you see in this PowerPoint and lecture are the ancestor of animals, fungi, and plants. Their ancestor and ancestor of the plants are the same. But the cells that are living today, the protists that are living today, are as evolved as anything else, right? Remember that. So this is really important. Algae, slime mold, protozoa, it's plants, fungal, animal cell. Those are the group that share a common ancestor, the most closely related group. The algae with plant cells. So protists with plants, we call the algae are the one that share a common ancestor with the plants. The slime molds and water molds are the ones sharing a common ancestor with fungal cells, and protozoa is the one sharing a common ancestor with animal cells. All of the protists and all of the eukaryotes share a common ancestor, right? Okay, so now how do we get there? How did we get to the point when we have that type of conformation? Well, it's all about that endosymbiosis. Remember how eukaryotic life started. From a common ancestor to all life, you have prokaryotic life with bacteria, and then you have archaea. One archaea, one lineage of archaea, engulfed a prokaryote, a bacteria. And that's when you have primary endosymbiosis. So you already had infolding here. Notice you already have a nucleus, right? So you already have a membrane-bound organelle. This is not anymore an archaea here. This is an ancestor of all eukaryotes. And then you have engulfing an aerobic bacterium and a cyanobacterium. That's why we are a green algae group here. Algae group, sorry, because you have a cyanobacterium and a aerobic bacterium, right? Gives rise to mitochondria and chloroplast. We don't show the other organelles, but they're there, right? So the first enfolding, the primary endosymbiosis, sorry, if it was a chloroplast that uses red, that actually looks red, so it doesn't use red, right? It uses blue and purple and other coloration for photosynthesis, it's the red algae. And then if it was looking green, not using green, but using blue and red, it would be green algae. Now, that green algae here, they became plant. We'll talk about that in the next short lecture video on plants. But green algae in and of themselves are right there. And the number of membranes around, you just want to zoom in here. That prokaryote here has one membrane. That prokaryote here has one membrane, and there is one from the ancestral eukaryote. Thus, when they fold over, you have two membranes now, and that's why your chloroplasts look with two membranes around. And your mitochondrion as well, they have one membrane and one membrane inside that's enfolded a lot. So two membranes around your chloroplast means that you are either red algae or green algae. Okay? That is, from green algae that got on land, you have plants, and we'll talk about it. That's the first step. We are now we are being in the eukaryotic realm, right? But then from there, any ancestral eukaryote, so another one here, engulfing a red algae, right? So the red algae, one of the lineage got engulfed by another ancestral eukaryote that doesn't have a chloroplast, just a mitochondrion. Then the rest of the red algae organelles kind of resorb, and all what's left is the chloroplast. And you notice that the chloroplast now has the two membranes, and there are two membranes now, one from the green algae, red algae, and one from the ancestral eukaryote engulfing it. So it could be four, or those two membranes can merge, so it would be a third one around the two. So it's 3 or 4 membranes, and that would be the brown algae. Okay? Now conversely, if that happens to a green algae, an ancestral eukaryote that's only heterotrophic with only a mitochondrion, engulfs a green algae, you get rise to the euglena. Euglena is a group that is heterotrophic and autotrophic. You can read more about it in the PowerPoint, but they turn on photosynthesis with their chloroplast whenever they don't have enough food in the environment, and they use the eye to orient themselves towards the light. And in this case, it's always 3 membranes. The membranes always have merged here in that lineage. That's what we call the secondary endosymbiosis. Now, interesting to think about, the red algae, they're really deep algae. They look red because they reflect it, right? And the reason why they reflect it is they don't use it. The reason why they're not using it, they're really deep, so the red wavelengths don't really get down there. So they need to use other wavelengths. And they only look red when you plug them out of a deep sea and put them out, because that's when the red gets reflected. Otherwise, there's no red deep there in the sea and the ocean. Brown algae, if you want to think about it that way, there's more membrane around, so instead of looking red, it looks a little bit more brown. It's not how it works, but it's a good way to remember it. So you must know the different endosymbiosis, primary, secondary. You must be able to identify your eukaryotic ancestor. They're having an endosymbiosis of a mitochondrium and a chloroplast. Cyanobacterium that will then become chloroplast. You must be able to explain red algae, green algae with 2 membranes and the 3 membrane from the brown algae and euglenas, right? This is a really important concept. Pay attention to the fact that it's an ancestral eukaryote that's engulfing a red algae to give rise to a brown algae lineage, or a green algae to give rise to a euglena lineage, right? Okay, so that's endosymbiosis, primary and secondary. Now, these, the cool stuff that they do, if we focus on the green algae, this will be coming back a lot later when I will talk about plants. So it's important to learn it now. This is the general reproduction cycle for a green algae. And it's really similar to plants. So notice different things before we dive into the details. There is a diploid phase and a haploid phase. That's called alternation of generations. This means that's like, imagine it's you. You have a zygote growing by mitosis, making an individual. Then that individual makes sperm or eggs by meiosis, but we don't go directly to fertilization. We have it's like the sperm or the eggs are growing into full-blown individuals, but they're haploid, right? So they have only one copy of all genes, one allele. So it's not that they're missing DNA, genetic material. They're missing a quantity of it. They have half of their genome gone, but they still have every genes. They just have one copy of it, right? That doesn't work for human beings. We skip completely that step of gametophyte, and directly the gametes reproduce and fuse. We go from gametes to fertilization. There's an entire step here when the spores grow up and then make gametes by mitosis, and those gametes mix and have fertilization. So we call the diploid individual the sporophyte. Sporophyte, the one that makes the spores. So sporophytes must be diploid because making spores is by meiosis, right? And the spores give rise to the gametophyte. The gametophyte is the one making the gametes. So that's the confusing part, because we are the gametophyte, we make gametes directly, right, by meiosis, but the gametophyte in plants and in green algae is the one making gamete by mitosis, right, because they're already haploid making a gamete by mitosis. Really important to chew on that and digest it very fully, because sporophyte, gametophyte, those terms are going to come back big time later. When we look at the slime molds and the ancestral of all fungi, it's a really interesting thing that's worth exploring a little bit for a second. Notice that they will just live their life, feed and divide here. We start down there. And then when there's a lack of food, they start to stimulate, aggregate, right? When they aggregate from that slug that's going towards light, and when it arrives in the light, it holds and makes a stalk with a fruiting body. And only the fruiting body individuals get to make spores that would germinate. Now, think about it that way. It means that all the individuals in the stalk and the fruit are perishing. They die. And they're not sharing DNA. They're just different individuals that come together to make that structure. And you may ask for yourself, why would they agree to die? It's like that their genes don't get passed on, right? Well, based on your understanding of evolution by now, you should have cracked the code here, and you should think about it that way. The only way we're seeing this is because they got together and did that, right? So that means everything else that did not do that died. And the reason is, when you lack food, if you don't make that body and that fruiting body, you cannot make a slug to move towards light. You cannot do all that as a single cell. So simply, the one that evolved, the population that evolved the capacity to do this, they survived. The other one, they died when there was no food. On top of that, the fruiting body and the stalk and the fruit or individual are all sharing the genes. Not all their genes are identical, but they're all sharing the genes that make the aggregation, the slug, and then the stalk. They're all sharing those genes. So those genes get passed on, because that is a successful behavior to actually get to make spores that would get to new habitats, because the spores can migrate via water, via wind, all of those, right? So all of a sudden you see the advantage that makes the individuals carrying those genes having a better, higher reproductive success. So we have differential reproductive success between all the slime molds. The one that do that behavior have a better reproductive success than the one that just die when there's not enough food. They don't go together. We have heritability of a trait, because the one that make those spores, they're carrying those genes. And we have variability of a trait. Some slime molds didn't do that, right? So we had evolution, and boom, population have evolved with that capacity to do that, and other population are just not there to tell the tale anymore. Right? So that's an interesting little thing. That's the ancestor of all fungi, mushrooms, if you'd rather think about it that way. So last thing I want to leave you with is the classification for protists is changing. I don't need you to remember that entire table, far from even remembering all the names. But notice all the different protists at the bottom here. We start to look more at the DNA, the genetic sequences, so what we learn in phylogeny, how we get to use DNA mutations to sequence. We sequence all those organisms and we classify them based on the number of mutations they share. Well, this is coming back here, and that's how we start to classify protists more and more. So we call supergroups now to stratify it a little bit more based on their DNA. But yeah, this is fascinating stuff. So if you're interested, you can dive into it. But remember, for your protist, the general picture is which one is ancestor of, like, I share a common ancestor with plants, those are algae, common ancestor with fungal cells, those are slime molds and water molds, and common ancestor with animals would have a protozoa. Make sure you know your endosymbiosis, primary, secondary, the green algae reproduction cycle that will come back for plants, and an interesting example of slime molds ingenuity. And I'll see you guys in the next one when we talk about plants.